Hi everybody, so today we're looking at fermentation, which is simply respiration without oxygen. So in fermentation we have the same first stage as we do in cellular or aerobic respiration, and that's glycolysis. So we want to know then, if we have glycolysis, what happens next? Why is it different? So if we're thinking about glycolysis, then of course we start with glucose, and then we're going to move to our fructose 1,6-biphosphate. That will then be split, so we'll have hydrolysis and we'll get triose phosphate, and then we get our pyruvate molecule. And of course this process includes a few other bits as well. So first of all, we need 2 ATP molecules to start with, which will get converted into 2 ADP plus PI. We also need 2 NADs, and they get reduced. And then our pyruvate molecule here, normally, if this were aerobic respiration, it goes off to the link reaction, and then it would go into the Krebs cycle. The other thing is that our reduced NAD normally goes to the electron transport chain, and when it's there, it will get oxidized back to NAD, which means that the NAD can then be reused in glycolysis. So we have this cycling around. The NAD gets reduced, goes to the electron transport chain, gets oxidized, comes back to glycolysis again. The electron transport chain is also producing ATP, which we can come and use at the start of glycolysis as well. There is other ATP made, of course, during glycolysis. I haven't included that there because at the moment that's not what is the most important thing. So if we've got no aerobic respiration, that means no link reaction, no electron transport chain, because now I want to think about what happens in fermentation. So we don't have these processes here, because you can't have those processes without oxygen. So if we don't have those processes, then first of all, what happens to the pyruvate? And of course, what happens to our reduced NAD? This is particularly important, because if the reduced NAD is not oxidized, we can't recycle it. And that means that this whole process here is going to stop, which means that we stop making pyruvate. Okay, so what we want to know then is what happens to the pyruvate. And first of all, we're going to look at what happens in mammals. So it's not just mammals, it's mammals and quite a lot of microorganisms as well. And the first thing, as we've just seen, so we've got glucose to pyruvate. So this is just a simplified version of glycolysis. And we know that the pyruvate is not going to go into the mitochondria and join the link reaction because there is no oxygen present, so that next stage can't happen. So in mammals, what happens is that first of all, we also know, as we've just said, that we've got to find a way to regenerate our NAD. We know as well that to go from NAD to reduced NAD, two hydrogens have been added. So if we want to regenerate our NAD, then in total we need 2 hydrogens to be removed so that we can regenerate our NAD. So we just need somewhere where we can basically donate hydrogens from. If this reduced NAD is able to give up its hydrogen, release its hydrogen, then it will be oxidized. So in mammals and lots of other microorganisms as well, the pyruvate is converted into lactate. It's basically the same thing as lactic acid, which you've probably heard about in relation to your muscles when they're doing anaerobic respiration. So this is the same thing. When that happens, to go from pyruvate to lactate, 2 hydrogens are donated from our reduced NAD. And that means then that we've then got oxidized NAD, which is regenerated. So by converting pyruvate to lactate, you recycle your NAD. There's an enzyme that's needed to turn pyruvate into lactate, and that's lactate dehydrogenase. Now, just something to be very careful of here. You need to know this name, but beware, because dehydrogenase normally is the enzyme that would remove hydrogen from something. But here, the pyruvate as we go to lactate, we're adding hydrogen. And that's just because this enzyme actually can help this to go both ways. So in different situations, you might have lactate turn into pyruvate. And that is a dehydrogenation reaction. So you don't need to worry about that, but just be aware that when we're going from pyruvate to lactate, we're using a dehydrogenase enzyme, but actually hydrogen is added to the pyruvate in that reaction. Okay, so what about yeast? Yeast cells and also some plant cells have a different process. So we start off in the same way. So we have our glucose undergoing basically glycolysis and we end up with pyruvate. And in exactly the same way, we've therefore got NAD being reduced and we need to find a way to oxidize it again. Okay, so we need to find something to give the hydrogen that reduced NAD has got. And in yeast, what happens is, first of all, the pyruvate is turned into ethanal. That releases carbon dioxide. But then we've got another stage. The ethanal is turned into ethanol, so it's an alcohol. And that is where we end up oxidizing our NAD. So this stage here is where the hydrogens from our reduced NAD are added, which then regenerates NAD. This reaction here is also catalyzed by a dehydrogenase enzyme, but this time it's alcohol dehydrogenase. One important thing to be aware of then, in this process here, we've still got a final electron acceptor. Even though we're talking about hydrogens, we've also got electrons being transferred. We just aren't talking about them. In aerobic respiration, oxygen is the final electron acceptor. Okay, but in fermentation, we have a different final electron acceptor. So in this example here, so in alcohol fermentation, because the reduced NAD, so here, this is where the electrons get accepted. So the ethanal accepts the electrons, because remember, hydrogen is made of protons and electrons. So the ethanol accepts the electrons and then becomes ethanol. So ethanal is the final electron acceptor. And if we go back to our lactate fermentation, the pyruvate is the final electron acceptor, because the pyruvate accepts the electrons from hydrogen and then we get lactate. So in aerobic respiration we have oxygen as the final electron acceptor, and in fermentation we have either pyruvate or ethanal as our final electron acceptor. Okay, just to finish, we want to know what happens to lactate and ethanol at the end. So basically, if we think about lactate fermentation, the lactate goes to the liver. Okay, this is the liver. And then once it's in the liver, it actually gets converted back in the opposite direction into pyruvate. And then once we've got pyruvate, two things can happen. If there's plenty of oxygen, then that pyruvate, which normally is, of course, is at the end of glycolysis, this pyruvate can just continue into the link reaction and then the Krebs cycle and the electron transport chain, and basically aerobic respiration can happen. So basically this will get oxidized into carbon dioxide and water. But under other conditions, so for example if there's not enough oxygen, then that pyruvate can just be converted into glucose. So again, this is basically going in the opposite direction. So this is going the reverse of glycolysis, and that glucose can then be turned into glycogen and it can be stored in the liver. So what's happening is energy that's in the lactate can still be used. It's not wasted, because by converting it into pyruvate, it can then be the energy can be released during respiration if there's oxygen, or by converting it into glucose, glycogen, again at some future stage, this can then be broken back down into glucose and respired. However, with alcohol fermentation, the ethanol, nothing can happen with it. the yeast, for example, is not able to convert ethanol into anything. So any energy that's left in here just gets wasted. Okay, that's it. Thank you.